Today I'm going to talk about Mucor rot, an emerging post-harvest disease of mandarin fruits in California. Previously, Mucor rot was found to be one of the 6 major post-harvest diseases of mandarin fruits in California. However, little is known about this disease and its causal pathogen on mandarin fruits. Objectives of the study are, therefore, to identify Mucor species based on genetic and morphological characteristics, and to examine their pathogenicity on mandarin fruits. First, sequence analysis was conducted based on partial ITS gene. Of 197 isolates, the most prevalent species was Mucor piriformis, accounting for 92.4%. Other minor species were 2 forms of Mucor circineroides, Form circineroides and Form lusitanicus, Mucor racemosus, Mucor hyemaris, and Mucor mucedo. Mucor species were also identified based on morphological characteristics by examining their structure. Here are some scientific terms. Tiny spores are called sporangiospore. When sporangiospores are attached to a structure called caramella, it forms a black sphere called sporangia. This whole structure is called sporangiophore. Here is a picture of sporangiospores, sporangia, and caramella of each Mucor species. Based on genetic and morphological characteristics, 5 Mucor species were identified, and their physiological characteristics were also examined. First, mycelial growth rate to various temperature was studied. Temperatures are on the x-axis and the mycelial growth rate on the y-axis. Red line is the growth rate of Mucor piriformis, the most prevalent species. The blue box indicates the storage temperature for mandarin fruits. At this temperature, Mucor piriformis grows 3 to 4 times faster than other Mucor species. Germination rate at 5 degrees Celsius was also examined. Various Mucor species are on the x-axis, germination percentage on the y-axis. Sporangiospores of Mucor piriformis germinate much faster than those of other Mucor species. Finally, pathogenicity tests were conducted. Organic mandarin fruits were surface sterilized by dipping them in 10% bleach for 2 minutes. After rinsed 3 times, fruits were allowed to air dry, and a wound was made on the skin, and sporangiospore suspension was inoculated on the wound. The inoculated fruits were incubated at 5 degrees Celsius. Disease incidence and disease symptoms were recorded. Here is the result. In this picture, fruits were incubated at 5 degrees Celsius for 16 days. All Mucor species cause Mucor rot on mandarin fruits to various degrees. Mucor piriformis appeared to be the most virulent species. Based on the previous studies about Mucor rot on pome fruits and stone fruits, here is a probable life cycle of Mucor piriformis in mandarin orchard. In spring, Mucor starts to sporulate on grass or chickweed. In summer, the fungus survives in decayed fruits on the ground or rabbit dung. The fungus can also infect stone fruits and sporulate during the summer. The fungus can be spread out in the orchard by birds, rodent, insect, or rain, and continues to infect fruits. Infected fruits could fall on the ground and thus spread out sporangiospores in soil, which could be further spread out by animals or rain. Infected fruits could also get in packing line and directly or indirectly result in Mucor rot during storage. During harvest, fruits beans are placed in orchard where they get in contact with infested soil, which could be later contaminate packing line. And finally, sporongia spores can overwinter in soil. The fungus can also survive in plant debris in orchard. Infested soil and plant debris on the ground are the major sources of inoculum. Removing decayed fruits or plant debris from the ground and preventing infested soil from getting in packing line could reduce mucor rot incidence during storage. Here is a summary. Mucor piriformis was the most prevalent and the most virulent species causing mucor rot on mandarin fruits in California. Orchard floor sanitation, prevention of soil contamination, and careful handling of fruits during and after harvest could reduce mucor rot incidence during storage. If you like this video, please like or comment, and don't forget to subscribe to my channel. Thank you for watching.